So first of all, thank you very much for this amazing recognition of my PhD. And I'm going to start very broadly. As human beings, we usually engineer things by designing a prototype, then building its model, meaning physical representation. And then by testing little by little what's wrong by this model, we come up with new engineering solutions. And nature also engineers, but differently, by introducing mutations randomly and then selecting for variants that are beneficial in a given environment. So, inspired by this approach, researchers can now improve the properties of molecules in the lab by mimicking the process of natural evolution. This means that, for example, if you have discovered a new fluorescent protein, but it's not bright enough in your favorite model organism, a way to improve its properties using directed evolution is by introducing mutations into the gene that encodes it, then expressing it, and then selecting for variants that have beneficial property. The power of this approach is that this landscape defined by the relationship between sequence and phenotype of the protein can be navigated towards local optima without being explicitly known in advance. And to illustrate the kind of challenges that I struggled with during my PhD, let's say you want to evolve a dynamical system. In this case, let's say you want to evolve a car, but you are starting from a car from the beginning of the previous century. So the question here is, what should the selection pressure look like for us to evolve this car starting from this car? Maybe one intuitive approach is to select for the speed of the car. However, if you select only for the speed of the car, you will probably end up with something like this. Meaning you will get the speed that you selected for, but you would lose controllability and many other benefits of the modern car. So if you look back over for the previous 30 or 40 years since directed evolution has been applied in the lab, the usual targets of this approach have been enzymes or antibodies or any kind of binders or fluorescent proteins. But on the other hand, in nature, we observe many other proteins that exhibit interesting properties. In particular, one interesting class is the one I refer to here as switchable proteins, which can exist in multiple different conformations, meaning they go large change of conformation, and also they are usually allosterically regulated. And what's particular about them is that they can exist in multiple states. Usually in off state, they do nothing, and then you can change their conformation with an inducer from the exogenous environment, and in this new state, they perform a certain function. And you can think of them very generally, so they really lie at the ability of cellular systems to respond to the changes in the environment, and they can be receptors sitting on the membrane, kinases, inducible transcription factors that respond to small molecules, or finally optogenetics which respond to light. And in my PhD thesis, I have worked on evolution of inducible transcription factors and optogenetic proteins. So the reason why these proteins have been relatively neglected in the past compared to the static or non-switchable proteins is because they're particularly challenging targets for evolution. And this is because there are additional requirements that need to be satisfied when evolving these proteins. In particular, there is a need to maintain the ability of the protein to switch through all of its conformational states and the ability to control the protein exogenously by the inducer. If any of those requirements is not satisfied throughout the directed evolution experiment, you basically end up with a useless protein, either a protein that cannot switch anymore or that is not exogenously controllable. So to illustrate what can happen, I'm here showing research from a recently published paper where researchers wanted to evolve a morphine-inducible G protein coupled receptor. And their strategy was to select for high activity of the protein while gradually decreasing the concentration of the morphine in the medium. And what they ended up with were these two variants in blue and in purple, which are indeed more active than the ancestral red, but it has practically lost their ability to respond to the inducer. So this would be the molecular analog of the rocket car from the beginning of the story. So to evolve truly switchable proteins, one can diversify the selection pressure, not only to select for the high on state, but also to select for the low off state, and then basically alternate between these two selection modes. This, as you can imagine, is very technically challenging and is usually achieved by coupling the protein to a fluorescent reporter, and then you have to, for example, sort on FACS first for high activity, then for low activity, and like that many times until you obtain a good switch. And in my PhD, I wanted to go a step further, and really the question was, how can we design an in vivo self-selection mechanism for directed evolution of switchable proteins? Or to rephrase the question, can we basically find a molecular pathway inside cells to which we can then couple a protein of interest, basically assign cells to evolve that protein, but in such a way that cells achieve both selection for high on state of the protein and low off state of the protein. And I'm going to jump straight to the core idea. we thought of using cell cycle as a system for achieving that kind of selection. We were inspired to use cell cycle because many of cell cycle components naturally oscillate during eukaryotic division. But the key question really here was which of the components, oscillations in which of the components are necessary for cellular proliferation. So we were, as a first part of the project, screening to different cyclins and cyclin CDK inhibitors to find one which would satisfy these following three requirements. If it's expressed in a pulsatil manner, cells would proliferate exponentially. If it's not expressed, cells would eventually stop dividing. And if it's constantly expressed, cells would also not divide. So these requirements B and C would mean that during evolution, if there are proteins that are assigned to control such a cell cycle regulators that are locked in one of the conformations, would basically be a dead end of the evolution because such cells won't divide. So we would select them out. To test this idea, I have engineered a panel of budding yeast strains, which was a good model for this question because we have studied cell cycle in this to death. And basically, I have assigned a light-sensitive transcription factor to control different cell cycle elements, meaning in each strain, different cell cycle regulators was screened for these three properties. And I have here used EL222, which is a light-sensitive transcription factor from the LAV domain family of proteins. And to cut a very long story of several years short, we have discovered that only cyclin CLIP5, which is an S-phase cyclin, satisfies these three requirements among the ones that we tested. And then to make the strains more amenable for directed evolution, we have also deleted the endogenous cyclins, which in part overlap in function with the CLIP5. This means that during evolution, they would not basically take over our exogenous control. And an interesting thing about this CLIP5 is that it was discovered right here before I was born. Actually, it was co-discovered by Naismith Group and by Cross Group at Rockefeller, which is where my mentor did his postdoc. So it was really here where the key element of my PhD originated. After designing these strains and finding this property of CLIP5, we were ready to perform directed evolution. And as a protein of interest, we used EL222, so this light-sensitive protein. And the beauty of using optogenetics for evolution is that in order to select for different phenotypes of interest, it suffices to simply change the light regime that you grow these cells on. So because now cell cycle is dependent on light, you can imagine that there is an initial light pulse pattern where all the cells proliferate happily, but then you can change the light pattern towards, for example, really short pulses to select for higher sensitivity in the protein, or for example, to more light to select for lower leakiness or lower basal activity of the protein in darkness. And because we were working with optogenetics, it was also interesting to try to change the color of light these proteins are responsive to from the original blue to green. So this is what we have done, and I will briefly guide you through our results. In the first campaign, we have grown cells under very short pulses of light, meaning 1 second of blue light given every cell cycle, which is around 100 minutes in budding yeast. And after the evolution, I have sequenced these EL222 regulators in cells, found many mutations, and then cloned out those mutations to another strain where they don't control the cell cycle to confirm that they're really causal for the discovered phenotype. And indeed, they were all more sensitive than wild type here in blue when exposed to light. And importantly for us, they all had retained the ability to switch off when the light was shut off. In the second campaign, we did the opposite. We wanted to kill leakiness of wild-type EL222, which was non-negligible. This was important, for example, for synthetic biology applications where you want to control a toxic gene, where you really want no expression in darkness. And we have discovered several variants which have very low leakiness indeed. In the third campaign, we have grown cells from under green light and discovered two different variants, which basically unlock the green responsiveness in these wild-type EL222, and which are only one mutation away from it. And moving forward, we were thinking what else can we evolve with our system? And the choice here was another optogenetic protein called 5-BPIF. 5-BPIF is based on phytochromes, which plants use to respond to light. It's a red responsive switch, and basically it's a two-component system where 5B and PIF interact under red light. The main disadvantage of this two-protein system is that it requires addition of an exogenous chromophore to work. So only plants can make this small chromophore, which 5B really needs to respond to light. And that means if you really want to use this protein in any other model organism, such as yeast, bacteria, or mammalian cells, you need to first incubate your cells with the chromophore and then perform the experiment. And this is the disadvantage that we wanted to tackle using directed evolution. So for that, we grew cells under pulsing red light in the presence of PCB, and then we switched the environment to pulsing red light, but without PCB. And in 10 out of 10 experiments, we have discovered a deletion of a yeast-specific gene called YOR1. This was quite surprising to us initially because we were wondering how can a deletion of a yeast-specific gene compensate for the loss of some plant-based molecule. So first step was to confirm that YOR1 deletion really allows these two proteins to interact under red light, and indeed it does so to the same extent as the addition of exogenous chromophore allows. And later we figured out the whole mechanism, which I'm just going to skip for the sake of time. But if you want to discuss this or use this system in your setup, please let's chat. And moving forward, we wanted to really make this method more general. And so far, I have shown you only the evolution of optogenetic systems. But in nature, many other switchable proteins are responsive to small chemicals and not light. So we were wondering how can we make the whole setup more generalizable. And for that, the strategy was to redesign the cascade inside budding yeast, basically to retain the light sensitive protein as an input to the system, which would allow us to control the selection pressure using light, which is quite neat, and then to have cell cycle control as the final output. Then in between those two, we can plug in a protein of interest, which can be a chemically responsive switch. And as a proof of concept, we have used RTTA, which is a part of a well-known Tet-on system, meaning a doxycycline or tetracycline-inducible transcription factor. So we have selected here for higher sensitivity of this whole cascade by growing cells under very short pulses of blue light. And after sequencing RTTA, we have discovered two different mutations, which indeed increase its sensitivity to doxycycline without compromising the off state, meaning the activity in the absence of doxycycline. So in summary, I have shown you a new method for directed evolution of switchable proteins, where the key idea was that cell cycle can be used as a selection mechanism to achieve both selection for high on and low off state of those proteins. And I have applied it to improve properties of three important molecular tools, sometimes coming up with unexpected solutions. And moving forward, we are excited about using this as a foundation for many more interesting and complex phenotypes, in particular to evolve nanobodies or any kind of binders which can be modulated by a light responsive domain, or magnetogenetics where we can control different optogenetic domains with light, and finally to evolve not only single proteins but whole networks of genes, and in this case apply it to tune the dynamics of the cell cycle itself, either towards faster cycles or slower cycles in budding yeast. And with that, I would really like to thank my lab, especially my PI, Sahand Rahi. We are a relatively fresh lab. I was the second PhD student to join. It was an amazing experience so far. And also we have openings for PhDs and postdoc to continue working on this line of research. So please get in touch if you are interested, either with me or by emailing Sahand. And with that, thank you very much for your attention.